Okay. Okay. Welcome to our session on cellular form and function. So we have done the reductionist. You guys are surprised we're starting all over. You know, it's nice to start the movie all over again. Yeah, it's exciting. Okay, but we're gonna zoom through the concepts because we already did them. But I'm gonna post it for you online if you need more refresh. Okay, cellular form and function. Okay, so we agreed that our cells look different because they have different structures, and they have different structures because they have different functions. And in order to perform their function, to perform their different functions, they need different structures. And how do we achieve that? We achieve that through, yes, by transcribing certain regions in the DNA known as the euchromatin, and each euchromatin is unique to a certain set of proteins that will give the cell the defining features. We looked in the microscope and we saw different tissues of cells, some of which are squamous, others are cuboidal, others are columnar. But the point is, they look structurally different because they have to fulfill different functions. They look different anatomically because they have different physiology. And that was achieved by the transcription of different regions that give you different structures to achieve different functions. These are the basic cell types. There was a question about that on the exam. I took it out. They had you shown a picture like this, and you had to tell which one is stellate. You guys cannot see? No. And you're just looking. Tell me, we cannot see. I cannot read the minds. So they wanted you, and you guys are just looking at me. So how do I remove that then? Okay, it's gone. Sometimes in life you just gotta wait, things will go away. Okay, squamous. So they wanted you, like for example, on the lab exam, you were going to be shown this picture, and you told, what is that? Is that stellate form? Is that discoidal? So you had to know which one is which. So for the time being, I'll eliminate that. It's too much memorization. But next exam, I'll bring it right back up on the final. Now let's just get rid of exam one. Okay. Um. Okay. So cell shapes and sizes. So most human cells are about 10 to 15 micrometers in diameter. So 1 micrometer or micron is 1 millionth of a meter. So there's a limit on the cell size. An overly large cell cannot support itself and may rupture. And we talked about that changing the dimensions of the cell will lead to changes in the volume and the surface area. And we said there are physiological processes that are dependent on surface area, such as why are we so careful with not changing the dimension of the cell? Why are we so careful with not changing the volume of the cell too much? Why are we concerned with hypertonicity, hypotonicity? Why? Excellent, excellent, excellent. So the diffusion is dependent on many things, like how small you are. The smaller the particle is, the faster the rate of diffusion. The more the surface area, the faster the rate of the diffusion. That's why your intestine has a lot of microvilli with really high surface area, and that's how we are able to achieve this absorption of nutrients from our digestive system into the circulatory system to be delivered to our tissues. And then we talked about magnification versus resolution. Magnification is being able to see tiny things that couldn't be seen with the naked eye. And then resolution is when you can see in finer detail. So, what are the three major components of a cell? The three major components of the cell are the three big ones are the nucleus and whatever that's inside it, then the cell membrane and everything that's inside the cell membrane, like the lipids, the phospholipids, the cholesterol, the transmembrane proteins, and the peripheral proteins. So we don't just study the nucleus. Don't just say nucleus. Nucleus, everything inside it too, like the nucleoplasm, the nuclear envelope, the nucleolus, the DNA. That's all inside the nucleus. Then the cell membrane, not what's below it, what's in it. Phospholipids, peripheral proteins, transmembrane proteins, the gated channels, the carriers, and then you have the cytoplasm and everything that's in it. Also, the cytoplasm has your cytosol, it has your mitochondria, it has your organelles, the ribosomes, it has your endoplasmic reticulum, and we said that the rough endoplasmic reticulum is different from the smooth endoplasmic reticulum because it has ribosomes embedded on the rough endoplasmic reticulum, making it rough surface. And we said there are freely floating, there are freely floating ribosomes. Why are there freely floating ribosomes? So that's the general job of a ribosome. A ribosome translates MRI into proteins. But we have ribosomes attached to the rough endoplasmic reticulum and ribosomes freely floating. So why is that? Can you please raise your hand and speak in complete sentences and loudly so I can hear you? Yes. The ribosomes and the rough endoplasmic reticulum. Don't say that. Say the rough endoplasmic reticulum.
They make proteins that go outside the cell.
Excellent. They be exported. Yeah, good. With a barcode. If you remember the name of the barcode, I'll give you extra credit on the exam. What's the name of that? I said it already in that room. What's the name of that barcode that we stick on it? 5, 4, 3, 2, 1, 0. You lost. You see, it pays to be businessmen and women. Pay attention. It's called signal peptide sequence. SPS, the signal peptide sequence. That is an additional sequence that the Golgi body, when it's, the Golgi body is the one like putting in box, the Amazon box, boxing in the warehouse. That's what the Golgi body does. Packages the proteins. When it sees that barcode, that there is signal peptide sequence, it's going to throw it outside, meaning that this is going international shipment. This is going outside the cell. But if it's a free-floating ribosome, then it makes proteins for ourselves, for the cell, locally, to be used. The cell needs proteins too, needs enzymes too. So we study the cytoplasm, and then there is the extracellular fluid, and that is located outside of the cell. So the extracellular fluid includes any fluid outside of the cell, including the tissue, interstitial fluid, the blood plasma, the lymph, and the cerebrospinal fluid. Okay, the plasma membrane. So the plasma membrane, we defined it as having 2 leaflets, and it appears as a pair of dark parallel lines, and it has an intracellular phase and an extracellular phase. So the membrane is like a lipid. It is abundant in lipids, has about 98% of the membrane as a lipid, okay? And the phospholipids are 75%. The phospholipids are 75% of the membrane lipids, and they are amphipathic molecules arranged in a bilayer. And there are hydrophobic phosphate heads that face the water on each side of the membrane. What about cholesterol? Cholesterol is about 20% of the membrane lipids. It holds phospholipids still and can stiffen the membrane. Glycolipids. Glycolipids are 5% of the membrane lipids. So we have glycolipids and we have glycoproteins. So sugar can stick to the top of the lipid in the bilayer leaflet, and sugar can stick to the top of those peripheral and transmembrane proteins. This, I cannot emphasize enough, this fluid mosaic model. This shows you in blue your cholesterol. How does cholesterol affect, how does cholesterol impact? How does cholesterol impact the excellent, makes it firm, excellent, stern, excellent, makes what sperm, makes what stern? The plasma membrane, excellent, excellent. And we said we have two types of proteins, one that's all the way either at the top leaflet and one that's either in the bottom leaflet. What do we call them? No, these are not transmembranes. These are the opposite because they are on one leaflet. Yes?
Glyco?
The proteins. Glyco is like when a sugar is sticking to the top. We're not talking about sugar. We're talking about proteins. Proteins that are either on the top leaflet or the bottom leaflet. These are peripheral because they are either on the top periphery or the bottom periphery. And then you have proteins that span both layers of the membrane. These are known as transmembrane proteins. Okay, so the membrane proteins constitute 2% of the molecules, but 50% of the weight of the membrane. So they are really heavy. So transmembrane proteins pass completely. This, I really need you to appreciate that. You call them transmembrane because they span the entire both leaflets. And they have carbohydrates attached to them. And please remember that this transmembrane region has a top part that is hydrophilic because it deals with water, remember? So the top part of the transmembrane is just like the membrane. The top part of it is touching the water, so that's hydrophilic. The bottom part of it is touching the water also, that's hydrophilic. The one that's in the core, in the middle, that's the one that's hydrophobic. So functions of membrane proteins. Functions of membrane proteins. So the membrane proteins, we have receptors, and receptors bind chemicals. Receptors bind chemical signals to trigger internal changes. They bind chemical signals to trigger internal changes, and they may cause production of second messenger within the cell receiving the message. So there could be receptors, they could be enzymes. So these transmembrane receptors, they could be these transmembrane proteins, they could be receptors, they could be enzymes, they could be protein channels. And they allow the hydrophilic solutes and water to pass through the membrane. So some are always open, called leak channels. Leak channels. So they are always open. That's why they call them leak channels. And then there are gated. So they're not closed all the time, but they're not open all the time either. They open based on a reason. The first reason is ligand. Ligand is a signal, meaning when a hormone binds to them, they open. Or when a stimulus binds to them, they open. Other type of ligand-gated channels are based on voltage. So if you have electrical activity, if the electric potential changes, now you don't need a hormone. Just the electric activity, the depolarization of the cell membrane causes the opening of these gated channels. Mechanically gated channels. So the mechanically gated channels do not respond to like a hormone. They don't respond to a voltage change. They respond to mechanical, physical pressure, stress. So if you touch them, then a lot will influx. Then they will open, allowing the influx of, allowing the influx of the ions. They are channels for what? For ions. So when they open to allow ions to enter or exit. So who makes them open? Give me one of the three reasons why would they open. These membranes, they are gated. So you know a gate cannot be opened for no reason. You need a reason to open it. Yes. Yes, give me a reason why it would open to allow the influx. What are the things that could cause a protein channel to open and let the gate open and let in? Yeah. Yeah. Excellent, excellent. So that we call that ligand. So it's good that we understand the concept, like the right particle touches outside, but we have our own language. So we say ligand gated, ligand. The particle is known as a ligand, as a stimulant, because it targets that particular protein channel. Another reason why the channel gotta open beside the right particle or the right ligand binding. What's the other reason? What's the other reason? I said it right now. What's the other reason? What is the other reason? Yes. Yes. Voltage. So if the electricity, the membrane potential of the cell membrane changes, then the gate is going to open. What's the other reason why the gate is going to open? Kurt? Yeah, mechanical. So when you physically, that's how when you touch, you feel the pain because they have been stretched, then they open to allow ions. So they're gated, but when they open, they allow ions to flow inwardly. So the functions of the membrane proteins, we have carriers that they bind solutes and transfer them across the membrane. We have pumps, and these are carriers that consume ATP. So there are cell identity markers. These are glycoproteins acting as identification tags. These are glycoproteins acting as identification tags. And there are cell adhesion molecules, and they mechanically link the cell to another cell and to the extracellular material. So you see, that's the chemical messenger binding to a receptor, and then a receptor that binds to the chemical messenger, such as hormones sent by other cells. So these are I think that's on the exam, where you're going to be showing this diagram and you're going to be able to tell which one is the receptor. So that's the ligand. The red one is the ligand. So the ligand binds the receptor. Here, that's an enzyme. The enzyme breaks down its ligand, and this is a leak channel. You see, it's always open. It's not gated. But this one is gated. It's closed, and it's open because of voltage or the right ligand binds to it. Then there's cell identity marker. The cell identity marker is a glycoprotein acting as cell identity marker, distinguishing the body's own cells from foreign cells. Why is that important? For immunosurveillance. So when your immune system is scanning yourself to tell self from non-self, it scans for those cell identity markers, these glycoproteins that tell immune system, don't kill us, we are self. Hands up, we are self. We are not non-self. And then there are the CAMs. The CAMs are cell adhesion molecules. These cell adhesion molecules bind one cell to another. Then we have second messengers. Why do we say we need second messengers? Why can't the ligand or the hormone go cross all the way through the plasma membrane, cross through the cytoplasm, bind the nucleus, and all of that? Why? Yeah, it's excellent. It's lipophobic. So the hormone is lipophobic. It cannot melt in the butter, that yellow region, that lipophilic region. So it needs somebody else. It will bind from the outside, but it cannot enter. But somebody can go in and do the terminal or target effect. Excellent. And a perfect example is what we mentioned here. You got to know this. You got to know this because this shows you how a second messenger works. So what's the first step? The red triangle. You see this red triangle is the first messenger. It binds the green receptor. Then the green receptor binds the green receptor binds the first messenger, and then the receptor releases the G protein, which then travels freely in the cytoplasm and can go on to step 3 or having various other effects on the cell. Then a messenger such as epinephrine would bind. Then the receptor releases the G protein, which then travels freely in the cytoplasm and can go on to activate other enzymes. So the G protein binds to an enzyme called adenylate cyclase in the plasma membrane. Adenylate cyclase converts ATP, adenine triphosphate, to cyclic AMP. So adenine monophosphate. Then cyclic AMP, that's our second messenger. Cyclic AMP would convert inactive kinases into activated kinases. And then the kinases would add, by phosphorylation, add a phosphate group to other cytoplasmic enzymes. This activates some enzymes and deactivates others, leading to varied metabolic effects. So cyclic AMP activates cytoplasmic enzymes called kinases. Then the kinases would phosphorylate the inactive enzymes, causing them to be activated and cause various metabolic effects. This is so important. I'm going to do it again. Oh boy, God help me. All right. This is very important. Why do we need second messengers? We always need second messengers when we can't get in. We cannot. When we were steroid hormones, lipophilic hormones, we can enter. There's no problem. Lipophilic hormones can cross the cell membrane. Lipophilic hormones can cross the cytoplasm and they can bind to receptors in the cytoplasm. They can even cross to the nucleus and they can bind to receptors on the DNA of the nucleus, then do the central dogma of biology, which is transcription of DNA into mRNA, then translation of mRNA into target proteins that are structural or functional. But now we don't have all the hormones lipophilic. We have some hormones that are lipophobic, so they cannot enter. So the lipophobic hormone binds to the outside. It could not enter the cell. It cannot traverse the bilayer leaflet. It cannot enter. So now it will bind on the outside. It bounded on the outside. It will cause a conformational change in the transmembrane protein. That's going to be a receptor. Then the receptor is going to cause G protein to go on and activate another enzyme that is embedded in the cell membrane, known as adenylate cyclase. And adenylate cyclase is going to do something very important for us, and that is it's going to convert ATP, known as adenine with 3 phosphates, triphosphate, is going to convert adenine triphosphate into AMP. So it used to be adenine with 3 phosphate. Now it's going to be adenine with 1 phosphate. Now it's adenine monophosphate. It was tri, now it's mono. Adenine triphosphate is going to become adenine monophosphate. This adenine monophosphate is going to be, actually, this enzyme known as adenyl cyclase, it cyclicizes. So the cyclic AMP, so AMP is not going to be just AMP, it's going to be cyclic circular AMP. This cyclic circular AMP, that's my second messenger. That's my second messenger who will mediate the effects of the first messenger who couldn't go in, because it is not lipophilic and it is hydrophilic, because it couldn't go in. So the second messenger, cyclic AMP, now is going to activate for us the inactive kinase is going to become active kinase. And the active kinase now is going to switch for us. The active kinase is going to switch for us an off enzyme that's in off, an enzyme that is closed, an enzyme that is off, to an enzyme that is on, and then the enzyme is going to give me my target effects. So now you see all of this. This is called intracellular because it's happening inside. Intracellular signal transduction cascade. This is called cascade. This one, A, pass to B. B pass to C. C pass to D. D pass to E. E pass to F. F pass to G. G gives us the final response. So you see like a domino. It's like a cascade. So they call it intracellular signal, intracellular, because it's inside signal transduction cascade to mediate the effects of this first messenger who cannot get in. It's like somebody who doesn't have ID to get into school. They outside, they tell somebody, the security guard goes to this guy takes up the elevator, goes to the faculty room contact the dean goes to blah blah blah, then he goes to this, goes to the president, then so on and so forth. So you see, this is the signal transduction pathway with the second messenger. But if you are lipophilic, that's like you have the ID. Lipophilic, you can just cross through the lipid bilayer. Clear? Any questions? Okay, that's good now. So now did we see that? Did you see this cascade? Okay, now the glycocalyx. We said that the glycocalyx is a layer of sugar where you have sugar binding to lipids on the membrane, plasma membrane, or sugar binding to proteins on the membrane, and that leads to I'm sorry? Glycoprotein, glycolipid, we screen them. That is like our fingerprint. That's what makes you self. That's what makes you recognizable. So why do we have glycocalyx? What are glycocalyx? Glycocalyx are carbohydrate moieties of glycoproteins and glycolipids external to the plasma membrane. So they are unique in everyone, but identical twins. So identical twins have the same glycocalyx. Now that sounds like a really good true or false question. Everybody has the same glycocalyx. True or false? False. No two persons can ever have the same glycocalyx. False. Who are the two that can only have who can share the same glycocalyx? A mother and her baby, identical twins, fraternal twins, a father and his son. Identical twins. No. Protection, immunity to infection. So that's why the glycocalyx is important for protection, immunity to infection, defense against cancer, transplant compatibility, cell adhesion, fertilization, embryonic development. Okay, so this is golden. I was saying this earlier. So now we finished with the membrane. Now we got to talk about the extension of the membrane. How many extensions do we have? We have the microvilli, we have the cilia, we have the flagella, and we have the pseudopods. These four, you got to know them. What they do. Microvilli are made up of microfilaments. That's an easy one. They are important because we find them in the intestine, brush border. And why is that important? To increase the surface area. However, if you look, that's them. That's the microfilaments, the brush border, and the glycocalyx that they have on your intestine, small intestine. Now let's look at the cilia. The cilia are hair-like processes, but the cilia, they are not for surface area. They cannot be found on your brush border. The cilia are for movement. So they move mucus down. When you go and all the mucus comes out, it is the cilia who moved it. It has two strokes, the power stroke and the recovery stroke. We're going to look at them now. So who enables the cilia to move? It is the dynein and the axoneme. You see, the axoneme is the core of the motile cilia, consisting of microtubules So that's the first distinction between microfilament and cilia. Microfilaments are for absorption. Cilia are for movement. Microtubules are in the cilia, while microfilaments are in microvilli. Microvilli are for absorption and increasing the surface area, while the cilia is for movement. The cilia has an axoneme where 9+2 structure. So there are two central microtubules surrounded by a ring of 9 parts. Then we have the dynein arms. The dynein arms curl up adjacent to the microtubule, bending the cilium and using energy from ATP. So cilia. So the cilia, they are based on microtubules, and the dynein, there's a dynein arm, and their central microtubules and peripheral microtubules. This is the ciliary action. So in the ciliary action, you see the power stroke. The power stroke when you move the mucus in one direction, then when you get up and stand up on your feet again, that's the recovering from the power stroke. That does not happen in the microfilament. That does not happen in the microvilli. That only happens in the cilia. There's also cystic fibrosis. So cystic fibrosis is a hereditary disease in which the cells make chloride pumps, but they fail to install them in the plasma. So that means you can never internalize and bring in chloride. So back in the day, doctors, when they wanted to find out if a patient had cystic fibrosis, they would lick the surface of the skin of the patient. If it is salty, that means the patient definitely had cystic fibrosis. They even used to go as far as tasting the urine to see if it had sugar. I'm sorry? I'm sorry?
After 1 year, were they doing that?
Salt? They were licking the surface of your skin. So if it's too salty, that means there is so much chloride, sodium chloride.
When were they doing that though? Like how long?
It was like, I'm sorry? No, it's not as like cystic fibrosis. So cystic fibrosis is much saltier. And same thing with urine. So, and then they also discovered that when a patient with diabetes urinates, all the ants crawl up to the urine because there's sugar in that urine. Okay, so doctors used to like go to far extent to like help the patient. So that's something we need to respect and appreciate. That, you know, nobody's like the doctor. I mean, tasting somebody's urine to see if they have diabetes. I'm glad I wasn't a doctor at that time. But it doesn't mean there aren't things that are as challenging that are nowadays. But I mean, yeah, that's the part of the evolution of medicine. Okay, flagellum. That is flagella similar to cilia or microvilla? Yes. You over here, your name is? Yeah. Okay. Is flagella similar to cilia or microvilli? No. Microvilli have, can you see the slides? So microvilli have, excellent. They have microvilli contain actin filaments. These actin filaments, they are the ones who make up the microvilli. You see actin microfilaments. But the cilia, they have, what do the cilia have? They have microtubules. Where are the microtubules? There we are. The cilia, you see the cilia has microtubules. Now, when you look at the flagella, what does the flagella have? The flagella have identical to cilium, whip-like structure with axoneme, just like the cilia had axoneme. You see the cilia had an axoneme of microtubules. Now the flagella also has an axoneme and it has microtubules. So that makes the flagella similar to cilia. Pseudopods. Pseudopods are continually changing extensions of the cell that vary in shape and size, and they can be used for cellular locomotion, capturing foreign particles. So the locomotion, so we're going to go and hug all of these all of these microbes and internalize them. And we're going to phagocytose them as phagocytes. So how do phagocytes work? We illustrated this first day of class. They go surround and then imbibe, bring them into a vesicle, like in a balloon, internalize them, then fuse them with a lysosome. So now you're going to have a phagolysosome. And then the phagolysosome is going to destroy them. And then the phagocyte is going to excrete them as edible blebs, edible, useless blebs that cannot harm. But how did the macrophage get over there? How did it's by locomotion and by pseudopods activity. Now, the plasma membrane and organelle membranes are selectively permeable, allowing some things through but preventing others. So we have two types of mechanisms. Okay, 1 second, please. There we go. Okay. Varicose vein from standing for too long.
Oh, you don't have some.
Oh, wow. Now that's much better. I should have thought of this earlier. Oh, wow. So now we're going to go on for 2 more hours. No, I was just saying that. I'm just playing with you. Okay. We're not going to go for 2 more hours. Okay. Okay. Plasma membrane. I look like I'm just standing. See, sometimes a little bit of thinking solves a lot of things. Okay. Plasma membrane and organelle membranes are selectively permeable. What selectively permeable means? It doesn't mean they are impermeable, but they discriminate who can get in and who can get out based on, not based on skin color, but based on chemical structure. What's happening in the back? So the plasma membrane discriminates who can get in, who can get out based on what? On chemical structure. Are you hydrophilic? You can't get in unless you have a transmembrane you can go through. Are you lipophilic? You can go in anytime you want. So that, and now the passage, allowing some things to go through, but preventing others from passing. So, passive mechanisms require no ATP. So passive mechanisms require no ATP. They don't require energy in the form of chemical energy. But does that mean they don't require energy at all? You can never have movement without energy. Maybe you're not moving based on chemical energy, ATP, but you're moving based on some other type of energy, which is who's powering the movement of diffusion? When we drew that beaker with 2 sides, one side 500 millimolars of glucose, and 100 millimotors of glucose, and glucose moved from the 500 side to the 100 side. Remember that? Remember when the glucose moved from 500 side to the 100 side? So the concentration gradient was the driving force. It was the incentive. But who manifested that movement? The vehicle that caused it to move. It was the what? Excellent. The kinetic energy, the molecules bumping to each other. They're moving and bumping to one another, leading to the movement. So here in passive mechanisms, we have kinetic energy. We have any sort of, but definitely not chemical ATP energy. That's a very big question on the exam. That you must understand that passive mechanisms require no ATP. It is the active mechanisms that require the use of ATP. So active transport and vesicular transport. So the carrier-mediated mechanisms use a membrane protein to transport the substances across the membrane. Okay? Filtration. So particles are driven through the membrane by physical pressure. So what is filtration? Filtration is when you move due to physical pressure. When you have a filter and you put, you put like, tea, when you put tea on top of the filter and you put hot water on top of the tea and then the water starts to filter out. What led the water to come out? Who's pushing the water from top? Is gravity. Excellent. So that's an example of filtration, that the movement is caused by a force. It's not ATP. It's somebody pushing down the gravity. In your heart, so in your capillaries, when the blood is pumped from your heart and it's going to your tissue, once it arrives to your tissue, what squeezes it out? So now let's look at this. That's your heart. Okay, that is your heart. Pumping blood to your tissue. When the tissue, you will have capillaries with a lot of holes to make all the tissue, to give the tissue oxygen. And then the bad blood will return to the right heart. So when the blood reaches the tissue, what makes it come out? What makes it get out? The blood. It's the heart. The heart pumping, squeezing, and then blood gets here, gets out. That's the pushing. It's the pushing, that's the hydrostatic pressure. So now you have pressure causing the squeezing out and the pushing out of the blood. That's due to filtration. That's a perfect example of filtration. So example of filtration of water and small solutes through the gaps between the cells, or filtration pores in capillary walls. We did the filtration. These are the filtration pores in your capillary wall. Another example, we said between the cells. So water goes also between the gaps of the cells. This is known as transcytosis. Transcytosis. You see, that's what I drew up there. That is now representing for us filtration through the wall of a blood capillary. Filtration through the wall of a blood capillary. So the blood pressure in the capillary forces the water and the small solutes such as salts through narrow clefts between the capillary cells. Okay, so you got solutes, water, capillary wall, and red blood cells. You see, they're all squeezing and coming out due to what kind of force? Filtration, good. Now, simple diffusion is the net movement of particles from one place of high concentration to a place of lower concentration. So that is due to constant spontaneous molecular motion. Constant spontaneous molecular motion. So molecules collide and bounce off of each other. So substances diffuse down their concentration gradient from an area of a higher concentration to an area of a lower concentration. And that occurs in air, and water does not require a membrane. So the substance can diffuse through a membrane if the membrane is permeable to the substance. Simple diffusion. So simple diffusion is factors affecting the diffusion rate through membrane. That is high temperature, high motion of particles. So that's what we were saying earlier. What causes the diffusion to become faster? That would be temperature. When the temperature increases and the increased motion of particles, there's also molecular weight. Molecular weight is when you have larger molecules moving slower, and the steepness of the concentration gradient. So if you have 500 on one side, 100 on the other, compared to 700 one side, 100 on the other, or 900 one side, 100 on the other. The gradient is so steep. So the more steep the gradient, the faster the reaction rate. This slide is one of the most important slides, not just in this lecture, but in the entire course, both AMP1 and AMP2. You knowing what are the factors that make the diffusion faster and what makes it slower. The temperature, you heat things up, diffusion will be faster. You cool things down, diffusion will be slower. Molecular weight, the smaller you are, the faster you're going to diffuse. The steepness, if you have 500 glucose, so if you had something like this, let's say you had something like this. If you had something like that, if you had something like this, let's say you had a beaker and it's divided into 3 regions. One is 700 millimoles of glucose. One is 100 millimoles of glucose. And one is 300 millimoles of glucose. Then you open up the permeability, the differentially permeable membrane, and you add and you allow the flux. Where do you think is going to be faster? Obviously, from area of higher concentration, you're going to have from 300 to 100 B, and you're going to have from 700 to 100 A. Which one do you think is going to move faster? Going from 700 to 100 or from 300 to 100? Raise your hand if you think 700 to 100 you're going to go faster. Raise your hand if you think 700 to 100 you're going to go faster. Raise your hand if you think from 300 to 100 you're going to go faster. The gradient, they're all glucose. We're not dealing with like there's salt here. Maybe if there was salt in this side and glucose, obviously sodium chloride is much smaller than glucose. Sodium chloride is one sodium atom, one sodium chloride. I mean, that's tiny. But we're not talking about glucose, who's a big molecule. Both are glucose. But here the separation is 700 minus 100. The difference between those two is 600. And the difference between those two is 300 minus 100. That's 200. Where is it more steep? Is it steep here, 600 or 200? So it's going to go faster where? So I don't know why you guys think it's 300.
I was thinking more so like it was the molecular weight is lighter.
Yeah, but we have the same glucose, G. You see the G? The three of them are the same. G glucose, 700 glucose, 100 glucose, 300 glucose. Oh, now I see. So now you guys thought that 300 means how big glucose is. No, 300 millimoles means how much glucose we have. Oh, you guys don't even know the units. That was in lecture 1. So 300 millimoles, it means like I have 300 pounds of glucose. 700 millimoles, like I have 700 pounds of glucose. Okay? So that is not the size. That is the amount. That's the concentration. That's the concentration. Osmosis. Osmosis is the net flow of water through a selectively permeable membrane. Okay, through the selectively permeable membrane. So water moves from an area of higher water, lower solute concentration to an area of lower water, higher solute concentration. So water can diffuse through the phospholipid bilayer, but osmosis is enhanced by aquaporins, So the channel proteins in the membrane specialized for water passage. So cells can speed osmosis by installing more aquaporins. So it is a crucial consideration for the IV fluids. So there's osmotic imbalances underlie diarrhea, constipation, and edema. Oh, look at this. That's amazing. Let's look at that. Look at side A and side B. Is there a difference in the water concentration? There's 1 liter in A, there's 1 liter in B. You see the line is straight. What's the only difference now? Is this orange particle on side B. So you drop orange particles inside B, but the barrier is only permeable to who? No, it's only permeable to water, because you cannot be permeable to one side and not the other. It's because it's moving to here. It doesn't mean it's permeable to side A. It can move in both directions, but it's because there is orange on one side. That means orange cannot get to side A. So that means it's impermeable to orange, impermeable to the solute. But no problem. We learned that water is a versatile solvent. It moves from an area of low concentration to an area of what? High concentration of what? No, not of water. Moving from an area of a low concentration to an area of a high concentration of what? Solute. Excellent. Whew. Okay, osmotic pressure. Osmotic pressure is the driving force. So how much liquid do you have? So now the same example with this. So now you don't have the permeability to glucose. Here you have water in all sides, A, B, and C. And water is the same on the 3, 1 liter of water on each one. But on side A, you put 700 of glucose, but glucose cannot move. It's impermeable to glucose. Too small to let glucose get out. 1 liter of water here, 100 millimoles of glucose. 1 liter of water here, 300 millimoles of glucose. Who has a higher osmotic pressure for water to move? A. So all the water is going to be going from low to high. And the water here too. 300 is less than 700. So all the water is going to be going to 700. So there is osmotic pressure forcing you to go to the 7. That's the osmotic pressure. Okay, that's the one pushing. Clear? So when there's movement, there's always pressure. Even in sociology. Why do the people go to the nightclub? What kind of pressure is pushing them to go there? Peer pressure, but what do they get over there? It's the mating, the sexual pressure, right? Isn't that what people do? No? That's not why people go to the nightclubs. That's what my friends went for. I didn't go. I've never been, but never. But that's where my friend, they used to go all the time. And they failed their classes. They drive an Uber. I'm not going to post this lecture now. They're driving Uber now. So you got to focus on your, but what I'm saying as an example is if you're moving, then if you are moving, then there's a pressure behind your movement, whether it's osmotic pressure, filter hydrostatic pressure, whether it's osmotic pressure, hydrostatic pressure, and what else is there? Active pressure with ATP. There gotta be, you will never move unless there's pressure. Clear? Now, this is really important, the tonicity. Okay. Let's say I have a red blood cell. Let's say that I have a red blood cell. Let's say I have 3 solutions. Solution A. Now I'm going to say goodbye to this marker. I've said goodbye to 3 markers today already. 1, that's A. That's A. That's B. And that's C. C comes before A for all intents and purposes today. Okay. This solution has a lot of solute. The solute here is so high. And this is your red blood cell. This is your red blood cell. That's your red blood cell. That's your red blood cell. The solution here is so high. The solution here is equal to the osmolarity in this solution is so high. The osmolarity here is higher than the red blood cell. The osmolarity here is equal to the red blood cell. And the osmolarity here is less than the red blood cell. What's the effect? What's the effect? You have a solution, you are bathing a solution of water, it is not a solution unless you have water, right? We said a solution, and last week we did solution, colloid, suspension, and emulsion. You remember? Remember that? Okay, solution is water plus salt. Colloid is water plus proteins. And suspension is water plus really big particles. Suspension. And emulsion is when you have a liquid dissolved in a liquid, like fat in breast milk, or like oil in vinegar dressing of the salad. Okay? So what happens now if it's really hypertonic? What happens now? The water, what's going to happen to the water inside the red blood cell? Excellent. It's going to come out to dilute here from an area of a low concentration to an area of a high concentration. Then what's going to happen to the red blood cell? It's going to shrink. Here, when it's equal, the red blood cell will be the same size. But here, when the solution is really low, the water is going to move inside the red blood cell. It's going to become really like a balloon. It's going to burst. It's going to pop. So that is a hyperosmolar solution. That is an isoosmolar solution. And that is a hypoosmotic solution. Okay, no problem. Okay. So now you guys are able to tell. Hyperosmolar. What happens to the red blood cell? You see, that's when the red blood cell is so like a balloon. What was it put in? What kind of solution was the red blood cell put inside? Hypotonic, so water moved in. If it's still the same size, that means what happened? The solution. And the hypertonic solution, what happens? Carrier-mediated transport. So protein carriers in the cell membrane carry solutes into or out of the cell. But remember, when we said that the diabetic patient's urine has sugar in it, so when a diabetic patient urinates, all the ants are going to go to the urine for the sugar that's in the urine. So what happened to the diabetic patient is that in the kidney, there are reabsorption. There are reabsorption glucose transporters. They are glucose transporters that take glucose from the blood and take glucose from the filtrate, throw it back in the blood. But the patient has so much sugar, then the sugar there is transport maximum. So all the carriers are occupied. So now the sugar starts leaking out. So that is only the transport maximum. Please don't allow any picture, any figure to escape you. Make sure you understand all these graphs. Okay, there are three kinds of carrier proteins. There is a uniport, a carrier that moves one type of solute. Then there's a symport, the carrier that moves two or more solutes simultaneously in the same direction. And then there's the antiport that transfers everything in the opposite direction. So what do we have here? We have facilitated diffusion. What's the perfect example of facilitated diffusion? The perfect example of facilitated diffusion would be the solutes of glucose. Glucose is polar, it cannot go inside the cell, but there is a carrier that will actually physically carry it and make a conformational change, bring it inside the cell. So the solute binds to a receptor site on the carrier, and the carrier changes conformation. And the carrier releases the solute on the other side of the membrane. Okay, secondary active transport. That's really important. The secondary active transport is based on primary active transport. So what is primary active transport based on? What is primary active transport based on? ATP is based on ATP. So let's look at this diagram. That's a perfect example. Let's look down here to the sodium ATPase. The sodium ATPase uses ATP to send out sodium against its concentration gradient. Okay, this is really important. Please pay attention. This is really important. Okay, guys. On our membrane, we have a very important sodium potassium ATPase. What does it do? This is our membrane. Okay, the sodium potassium ATPase has 3 seats on one side, and it has 2 seats on the other side. What it does is sodium, potassium, ATPase. We know that there is a lot of sodium on the outside. There's a lot of sodium on the outside and a little bit of sodium on the inside. And we know there is a lot of potassium on the inside and a little bit of potassium on the outside. So what is the natural movement of sodium now, according to what we have been learning so far? Is for sodium to move from which direction to which direction? Good. Sodium move from outside to inside, or move from potassium move from inside to outside. What kind of movement is that? Diffusion. Diffusion, excellent. That's diffusion from an area of a high concentration to low concentration, high concentration to low concentration. Now look at what sodium potassium ATPase does. Sodium potassium ATPase will take out three sodiums outside. Send out three sodiums outside and bring inside two potassium. Now, isn't that interesting? So what is it doing now? And obviously, what is it getting paid to do? It's getting paid with ATP. It hydrolyzes ATP to ADP. So it hydrolyzes ATP to ADP in order to send out sodium and bring in potassium. So why is it doing that? So is it working down concentration or is it working against concentration gradient? Against it. Because the concentration gradient is for potassium to flow out. But it's bringing it back in. It's bringing it back in. And sodium is supposed to come in, but it's sending it back out. So what is sodium potassium ATPase doing? It's not regulating. What is it doing? By doing that, what is the sodium potassium doing? Yes, ATPase. It's ensuring that there is no equilibrium. If there was no sodium potassium ATPase and you had 144 millimolars of sodium outside and you had 10 millimoles of sodium inside, right? That's how it is. You have 144 outside, 10 inside. Here you have 160 inside millimoles and you have 9 outside millimoles. If there was no sodium potassium ATPase, they're gonna equilibrate, right? So if you had 500 here, 100 here, after a while it's gonna be 300, 300, gonna be 300, 300. But when sodium potassium does that, when these guys flow in, she send them back out. Flow in, she send them back out. Sodium potassium is ensuring that outside still remains more, even if so it's reversing the equilibrium, ensuring that there is always gradient, ensuring that there is an inequality, ensuring that there is a disparity. So why is that important? That is important for the action potential, as we will learn later on. That when it comes to the action potential, what happens is, in order for the action potential to happen, some sodium channels must open, and then sodium will flow from an area of a high concentration to an area of a low concentration. But a person who's dead, they can never have an action potential. Why? Because the electricity between outside and inside is at equilibrium. So if you open the channels of sodium, is for a dead person where they have 300 outside, 300 inside. If you open the channels, will sodium flow in? I'm sorry? Forget dead. Even if they are, if something is alive, you have 300 outside, 300 inside. You open the channel. Is there going to be any movement? No, because there's equilibrium. But when one is 1300 1 is 300, then you open, then they will flow down concentration gradient. So now, this sodium potassium ATPase is ensuring there is inequality. One is more than the other. It's ensuring that. So that when we have an action potential, the action potential says, we have reached the threshold. Let's have an action potential. Then the sodium channels will open. Oh, sodium is more outside. Sodium went in. That is called depolarization. So action potential is how we mediate homeostasis. Remember when we said in homeostasis we have a stimulus detected by a sensor, a detector. Then the sensor detector is going to send the input signal to the integrator. Then the integrator is going to send an output correcting negative feedback signal to the effector. Then the effector is going to mediate the effect of homeostasis. Then in the end, you are going to have homeostasis. And then you're going to stop. All that communication was based on action potential. I know we haven't studied action potentials so far. Action potentials work like this. So this is the time in milliseconds. This is the time in milliseconds. And this is the concentration. So what happened was, in order to depolarize, sodium must increase. And to repolarize, potassium will, so sodium channels will open. This is the membrane voltage. I'm sorry, that's not the concentration, membrane voltage. So when we depolarize, we increase the membrane potential by opening the sodium channels, and we repolarize by opening the potassium channels. We depolarize by opening the sodium channels. We repolarize by opening the potassium channel. We depolarize by opening the sodium channel. We repolarize by opening the potassium channel. We don't need to know the action potential now. But what we need to know, this depolarization here, the red depolarization here, will only happen when sodium flux inward, influx of sodium. And sodium will only influx if you have more sodium outside than inside. So a dead person is not because they're dead, but because their outside is 300 and inside is 300. So when the action potential says depolarize, sodium come in, it says, I cannot come in, we're equal outside and inside. Does it make sense? Okay, let me say, does it make sense? Okay, let's do it again. 300 outside, 300 inside. If we want to depolarize, we don't have to worry about an action potential for now. Just understand that for the action potential to happen, we need two phases. A depolarization phase, and a repolarization phase. Both of the phases are mediated by the opening of the channels. So the depolarization is mediated by opening sodium channels, and repolarization will happen when we open potassium channels. So depolarization will happen when sodium moving from a lot outside to the little inside. And repolarization will happen when the potassium flow out from inside to the outside. But in the end, in order to have that, sodium will never move in from outside to inside unless what? Unless there is more outside to force it. Excellent. Unless there is more outside to force it inside. Potassium is not going to flow outside, except potassium is never going to flow outside unless there is more potassium inside. So you're never going to move from one direction to the other unless you have somebody to power your movement, and you need to have a concentration gradient. So now, if there was no concentration gradient, if there was no concentration gradient, can sodium influx? If sodium cannot influx, can I have a depolarization? No. If potassium is equal inside and outside, can I have potassium efflux going out?
No.
So I cannot have repolarization. And if I cannot have depolarization and repolarization, it means I cannot have action potential. And if I cannot have action potential, I cannot communicate. How does the hypothalamus tell the blood vessel dilate? Action potential. How does the hypothalamus tell your sweat gland secretes sweat? Action potential. How does the pancreas detect there is a change, an increase in sugar and secrete insulin? Action potential. How do we move our arm? Action potential. Action, everything action potential. But we need that influx and efflux. And we can never have it unless we have a gradient. So now you see why the sodium potassium channel is so important. That's why when you get bitten by a cobra or a snake, the venom of the snake targets that digoxin targets the sodium potassium ATPase. It shuts it down. So now you will disrupt the gradient. You will be at equilibrium, and then you can never have action potential. So you can never restore homeostasis. Okay. Now you see the sodium potassium ATPase throwing 3. So you need to know it uses ATP number 1. It uses ATP number 2. It takes out 3 sodium, throw them outside, and it takes 2 potassium from outside, bringing it back in. You gotta know this, that the sodium potassium ATPase use ATP for active transport by sending things against the concentration gradient. And that's how useful it is. Can you see this diagram? This diagram is very special because look, at one side we have the siglet. What's the siglet? That's the sodium glucose transporter. So the sodium glucose transporter utilizes this. So you know, if you're going somewhere already, somebody can hitch a ride. They can ride with you, right? Somebody can take a ride with you. So what's happening in the siglet is that sodium already has an efflux gradient to move down the concentration gradient, okay? So the sodium can already move down, okay? While it's moving down, glucose says, can I come with you? And then glucose enters. That's why it's called sodium glucose transporter, siglet. So you see the siglet at the top. Who's green? Who's green? Green is glucose. Who's orange? Orange is sodium. So now sodium is entering. Glucose say, can I come with you? And they both enter together. When they enter, don't stop there. Look at what happens when they enter. Glucose enter, the cell uses it for metabolism. So now we get ATP. What about the sodium that enters the cell? You see what happened to the sodium ATPase? It takes it, throw it back out again. So when the sodium potassium throwing out sodium outside, it keeps removing the sodium that went in with the siglet. You get it? Do you get it?
Yeah.
So when the sodium enters, when this sodium enters, you open the sodium potassium ATPase. We're talking about this.
Oh shit. Sorry.
So when the green, that's why you gotta look there. When the green, that's the green, that's our sugar. And the orange is our sodium. So sodium is entering, that's no longer a secret, that sodium has a concentration gradient from outside to inside. There is a lot more sodium outside than inside. It's no longer a secret. So sodium is moving in. The glucose will say, can I come with you? Now they will both enter. When they enter, the glucose is already shuttled in the glycolysis, and now we got ATP. But what about the sodium that accumulates? The sodium potassium is waiting for it, says, I'll throw you right back out. So it keeps removing it. So no matter how much sodium goes in with the siglet, your sodium potassium ATPase removes it. So it's always creating a gradient for more sodium to come in, because the ones that come in, the sodium potassium ATPase is throwing them out. So singlet enters sodium with glucose through this door. Potassium ATPase will take the sodium, throwing it out, and making sure that it's empty. There's not a lot of sodium. Yeah, we said that. You see in that diagram, sodium is always a lot. Look at this diagram. Orange represents sodium. So you see there is a lot of sprinkled sodium up there. You see the sodium? A lot of sodium. And you see green, that's glucose. So now sodium moves in from an area of a high pressure to an area of a low pressure. So now when it's an area of higher concentration gradient to an area of a lower concentration gradient, so now the sodium moves in and the glucose moves in with it. That's an example of secondary active transport because glucose is moving in with the help, hitching a ride. Sodium is moving, glucose says, can I come with you? That's called secondary active transport because glucose is not moving by itself, it is moving with the help of sodium. Okay, when glucose enters the cell, we know what happens, catabolism, endothermic, exergonic, we used it, it's gone. What about the sodium who came in now? We're going to have so much sodium inside. Now, sodium, potassium, ATP says, don't worry, I'll recycle that. It will take the sodium from inside, throwing it back outside. That's why we will always have glucose coming in because the gradient, if you have a lot of sodium build up in the inside, you're going to have equilibrium. So now sodium cannot enter, glucose cannot enter. You're going to have deficiency in glucose. So that's why the sodium that enters the potassium ATPase, sodium potassium ATPase will throw it right back out. Then the sodium potassium ATPase, that's really important. Really important, you knowing the sodium potassium ATP is a transmembrane, use ATP, throwing how many sodiums? 3. From where to where? From inside to out or from outside to in? Nope.
Oh, throwing it back.
From inside to out. And potassium, how many? 2, not 3. Potassium, 2. From inside to out or from outside to in? From outside to in. Okay, and that shows you the different ways that we can internalize, our molecules. So we have, that's a phagocytic vesicle. Let's now look at a phagocytic vesicle. So the phagocytic vesicle in phagocytosis, there's intracellular digestion and exocytosis. Let's look at phagocytosis. We know that, what does phagocyte mean? Yeah, phagocyte means eating. Yeah, excellent. Galloping, swallowing, swelling, swallowing, engulfing. So a phagocytic cell encounters a particle of foreign matter, and the cell surrounds the particle with its pseudopods. And the particle is phagocytized and contained in a phagosome. It's contained in a phagosome. So the phagosome fuses with the lysosome and becomes a phagolysosome. So the phagolysosome now the enzymes of the lysosome digest the foreign matter, and then we're going to excrete it as edible blebs that are harmless to the body. So make sure you know every diagram is you got to know it. The steps of every diagram, what's happening. The diagram, like this one too. So now this is receptor-mediated endocytosis. So what happens here is extracellular molecules bind to receptors on the plasma membrane, and the receptors cluster together. So the more you bind, the more the receptors cluster. Then the plasma membrane sinks inward and forms a clathrin-coated pit. And that clathrin-coated pit would close and pinch off. So you would have the pit separates from the plasma membrane and forms a clathrin-coated vesicle containing concentrated molecules from extracellular fluid. Transcytosis. Transcytosis is the transport of material across the cell by capturing it on one side and releasing it on the other. An example is the receptor-mediated endocytosis moves it into cell and exocytosis moves it out. Who did I say is responsible for packaging like a warehouse in the cell to send out material outside the cell or repackaging? What organelle is responsible for that? I'm sorry? What's the name of the organ that is responsible of closing the Golgi body? Excellent. Exocytosis is the so endocytosis bringing in, exocytosis sending out. So exocytosis is the discharge material from the cell, and examples are the release of insulin by endocrine cells, and sperm cells release enzymes for penetrating egg, and the mammary gland cells release milk sugar, and also functions to replace endoplasmic membrane lost by endocytosis. That's transcytosis. When we move, that's pinocytotic vesicle. The release of insulin by endocrine cells, sperm cells release enzymes for penetrating the egg, and mammary gland secretes milk. Also functions to replace endoplasmic membrane lost by endocytosis. Okay. Capillary endothelial cell. Now let's look at exocytosis. So exocytosis, you would have a dimple, and the dimple is a plasma membrane linking the proteins, and there's a secretory vesicle. This is a secretory vesicle. And so in exocytosis, you would have a secretory vesicle. The secretory vesicle would approach the plasma membrane and docks on it by means of linking proteins. So there's no receptor here. And the plasma membrane caves in at that point to meet the vesicle. So you see what's happening here is different from a clathrin-coated pit. You see in the clathrin-coated pit, we're trying to enter from outside to inside. We bind the receptor and then we get internalized. But in exocytosis, it's the opposite. We are moving out. So now the secretory vesicle approaches the plasma membrane and docks on it by means of linking the proteins. So the plasma membrane caves in at that point to meet the vesicle. So the plasma membrane and vesicle unite to form a fusion pore through which the vesicle contents are released. So the cytosol is clear, viscous, watery colloid. So what do we say colloid was? What's the difference between a solution, suspension, colloid, and emulsion? Solution has water and little small tiny particles like salt or sugar. A suspension is when you have plasma with water along with red blood cells. And then a colloid is when you have water with proteins. And what's an emulsion? An emulsion is when you have a liquid dissolved in a liquid, such as oil, vinegar. Oil, vinegar, like the salad dressing. Structural support determines the cell shape, organizes the cell contents, and directs movement of materials within the cell, and contributes to the movement of the cell as a whole. It is composed of microfilaments, intermediate fibers, microtubules. So that shows us the cytoskeleton. That shows us the skeleton of the cell. That's why they call it the cytoskeleton. You have desmosomes that connect like junctions. They hold together, they hold cells together. And you have secretory vesicle in transport. And then there's a lysosome. And then there's a terminal web, microfilament and microvilli. So centrosome, these are centrosomes, these are microtubules, and these are hemidesmosomes. Components of the cytoskeleton, you have microfilaments, thin filaments, and they form terminal webs, and there's intermediate filaments, and there's microtubules. Microtubules are protofilaments. So you see the protofilaments, the dynein arms, and the microtubules. Organelles, we have different organelles. You have these organelles, you can read them at home. But remember that I think there's a question on the exam about which organelle is responsible for programmed cell death. What is programmed cell death? Yeah, very good. So the cell has to program its own death, like suicide. So suicide of the cell. Who manages the death of the cell? By itself. She kills itself. And she kills itself, why? Because you have misfolded proteins. Proteins that are misfolded, they pile up inside the cell, so to cause damage, like mad cow disease. You have a lot of misfolded proteins on the brain cell that causes mad cow disease. Or for heroin addict, cocaine addict, they have a lot of problems with protein folding, so now it's going to start accumulating in the brain. They're going to have mad cow disease. And when these proteins are damaged, they get tagged. They get a tag like a postal stamp that says you got to be degraded. You know the shredder, the paper shredder? Now, there's a shredder for proteins known as the peroxisome. So the peroxisome is not the peroxisome. Yeah, the peroxisome. The peroxisome is able to shred down all the proteins that are damaged. You see, that is your peroxisome. That's the peroxisome. So if there is a misfolded protein that must be damaged, you see the purple tag is called a ubiquitin tag. A ubiquitin tag will be tagged on the protein that is misfolded, that needs to be shredded and destroyed. And then it goes inside the proteasome and it comes out as degraded protein. Okay? Proteomes, these are the proteasomes. They are hollow cylindrical organelles that dispose of surplus proteins. And they contain enzymes that break down targeted proteins into short peptides. Okay. So these are the organelles. We said that the mitochondria is important for programmed cell death. So if a cell needs to die, it is the mitochondria who will initiate the programmed cell death of the cell. So the cell will die because of the mitochondria. That's the nucleus. It has chromatin inside, euchromatin, heterochromatin, and you have endoplasmic reticulum. These are the network of interconnected membranous channels called the cisterns. And there's rough endoplasmic reticulum. We talked about that. It has the ribosomes that's making proteins that are going outside. And the smooth endoplasmic reticulum, it has the smooth lipid synthesizing and toxic detoxification. Ribosomes, we know those, they are translating proteins. These Golgi bodies, they are a system of membranous cisterns that synthesize carbohydrates and modifies newly synthesized proteins. So they are vesicles. So they are secretory vesicles. You see the cisterns, and they make these balloons, these vesicles for packaging. Lysosomes, that's what you were talking about earlier. Lysosomes are important because they are for degrading, lysosomes are important for degrading cells that are dangerous, like your bacteria, like the infectious microbes. They get destroyed by the lysosome. What are peroxisomes for? Peroxisomes. What are peroxisomes for? Nope, they're not for programmed cell death. That's the mitochondria. The mitochondria is the one who initiates the death of the cell. What are peroxisomes for? Yeah, excellent. To shred the proteins that are damaged. You see the purple protein? You see over there? That protein is misfolded. It's going to give us mad cow disease. So we need to tag it for destruction. And we tag it with this purple tag. It's called a ubiquitinase tag. A ubiquitin tag. That is a ubiquitin tag. U-B-I-Q-U-I-T-I-N. Ubiquitin tag in purple. Whenever a protein has a ubiquitin tag, the proteasome is going to run and shred it. That's your mitochondria. as the powerhouse of the cell. It can make the cell survive with energy, and it can also kill the cell when the cell is badly damaged and it needs to be removed so another cell can be built in its place. And we have inclusions. These are like saturation. They could be good inclusions like granules full of glycogen, pigments, and oil droplets and fat tissue. And they are for storage, or they are storage products of foreign matter in the cytoplasm. They are never enclosed in a unit membrane, not essential for cell survival. Okay? All right, guys. Thank you very much. See you next week. I'll post up the